Today we are going to talk about cellular events which takes place during acute inflammation, right? Now, first of all, we will talk about process of margination, that how white blood cells or leukocyte from central axial blood flow, they come to the periphery, to the near the endothelial cells. We call this process margination, which is basically a result of hemodynamic alteration in the inflammatory tissue. So today I will explain this. After that, we will talk about process of rolling of leukocytes, especially neutrophils, along the endothelial cells, which is mediated by selectins, right? After that, we will go to the next area where rolling neutrophils along the endothelial cells will eventually stop rolling and firmly become adherent with the endothelium, and that process is mediated by integrins, right? And in the end, we will talk about that when neutrophil passed through the endothelial layer, either in between the endothelial cells or through the endothelial cells, right, escape the endothelial layer, and from the lumen they transfer to the outside of the endothelial lining towards the basement membrane, and eventually they break down the basement membrane and come into interstitial area. There, in next lecture, we will discuss that eventually under the process of chemotaxis, neutrophil will reach to the focus of injury, and at the focus of injury, neutrophil will do phagocytosis and lysosomal and phagosome fusion and try to kill the bacteria, or they will attack the offending agent or microbes, pathogen, by the oxygen derived free radicals, or they can make a very special process of trapping the microbes by the process of formation of net. Net is neutrophil extracellular traps. But these are all different steps of cellular events of acute inflammation. Right now, let's focus on margination. Now, what is meant by margination? First, we should, to make the concept of margination clear, first we should know what is the blood flow through the circulation in a normal tissue which is not inflamed. And then I will tell you circulatory changes in the vasculature in inflamed tissue, right? And those circulatory changes and hemodynamic, how they kick the neutrophil from the axial central blood flow to the periphery towards the endothelial cell. So first of all, we see the normal blood flow, right? Let's suppose that this is arterial side, right? And let's suppose this is microcirculation, which is characterized by endothelial lining. Let's suppose these are postcapillary venules, and this is the venous side. Now what we have here, we have arterial side. This is suppose representing the circulatory system normal, and then I will compare it with the circulatory system in an injured tissue or inflamed tissue. Now in normal tissue, how blood is flowing through the microcirculation, we call normal blood flow laminar blood flow, laminar pattern of blood flow. Now what is laminar pattern? Laminar pattern of blood flow through the circulation of healthy tissue is that when blood is flowing, the particles like WBCs, red blood cells, and platelets. So particles are flowing in the center. We call it central axial flow. And plasma is more on the periphery, right? So cells are in the center and plasma is on the periphery, right? In a normal blood flow. And out of the particles, bigger particles are the most central. Let's suppose here I show normal blood flow and here are suppose white blood cells. These are white blood cells, right? Now white blood cells are usually in the center. Suppose neutrophils are usually in the center. Why they are in the center? Because they are the biggest individual particulate matter in the blood, healthy blood. Then on the periphery, we have, what are these cells? Red blood cells. Red blood cells are on the periphery, right? And most peripheral are, yes, what are these? Platelets. These are platelets, and most peripheral, just with the endothelial cell, this is plasma, cell-free plasma moving. What is this? This is cell-free plasma which is moving with the endothelium. Now you can see this, that blood components are moving in special layers. They are moving in special layers, and different layers are smoothly gliding on each other. And these layers and this way of blood flow, this is called laminar blood flow. This is how blood is moving. Again, I will repeat. First of all, first principle you should understand, in healthy tissue, laminar blood flow contains cellular component in central axial flow and cell-free plasma in the peripheral, most peripheral area in touch with the endothelium, right? Now, what happens in inflamed tissue? In inflamed tissue, this arrangement of laminar flow is disturbed. And what really happens in inflamed tissue, that neutrophils or white blood cells from the center, they are pushed to the periphery. I will tell you exact mechanism how they are pushed to the periphery. So in inflamed tissue or injured tissue, white blood cells are kicked out of the central flow and they are thrown to the periphery, or they are thrown from the central flow to the marginal flow. They come to the margin of the blood column. So they have more chances to hit and bump the activated endothelial lining, which is sticky, which will stick the white blood cells, which will be expressing adhesion molecules or their ligands or their receptors to make the neutrophils to hold it here. Now, in injured area, why we want to bring the neutrophil on the margin? Why? There's the reason. If neutrophil remain in the center, from arterial side, blood will keep on coming, and if the neutrophil remain in the center, they will keep on pushed. They will have less chance to touch the endothelium, and if they don't touch the endothelium, then they don't become sticky there, and if they don't become adherent with the endothelium, then they don't have a chance to migrate out of vascular compartment. Rather, if they stay in the center, they don't come on the margin, then as more blood comes, they will move through the microcirculation into venous side, and inflamed tissue will not be able to capture the neutrophils or white blood cells. So this process, getting the leukocyte from central axial blood flow to the marginal flow, or we say neutrophils were moving in the center, they are kicked out on the margin. This process is called margination, and this process occurs in microvasculature of inflamed tissue. But exactly how it happens? how margination occurs. Let me redefine the margination. Margination of white blood cells. Margination is an event in the inflamed microvasculature where leukocytes from the central axial flow are shifted in a lateralized position to the periphery towards the endothelial cell so that they interact, so that neutrophil has more chance to touch, hit, bump, kiss the activated, sticky, adherent endothelium, so that they stick with the endothelium and neutrophils are not washed out of the inflamed area. Now, I will make here inflamed area, right? Inflamed area number one, what will happen? The arterioles will undergo dilatation. The process is called vasodilation, right? Or to be more specific, we call it vasodilation. More specific, we call it arteriolodilation. First thing, there's arteriolodilation. And here I'm showing the microcirculation. And here I'm showing you the venous end. Now, if this tissue is inflamed, then what will happen? Suppose this is microcirculation in an injured tissue, inflamed tissue. As you know, that inflamed tissue, lot of chemical mediators are produced. Lot of chemical substances are produced locally, which will initiate and mediate the different steps of inflammation. For example, first step is that vasodilation will occur, right? And this vasodilation, I will explain how it contributes towards margination. Now, vasodilation, what happens due to vasodilation? There is increased blood flow. There is increased blood flow into tissue, right? And increased blood flow will lead to what? Yes, if there is more blood coming, then there will be increased hydrostatic pressure. Hydrostatic pressure. In inflamed tissue, when arterioles will dilate, more blood is coming, and when more amount of blood is coming due to arteriolodilation or vasodilation, then what will happen? That in microcirculation, whenever I use the word microcirculation, actually I mean here specifically I mean post-capillary venules, right? Now, in microcirculation, when more blood is coming, pressure will increase. What pressure will increase? Hydrostatic pressure will increase. And suppose these are endothelial cells. These are endothelial cells. When hydrostatic pressure will increase in this area, right, then the fluid which is inside, that will leak out, right, and that will leak out under high hydrostatic pressure. This fluid at very initial stage, this fluid which is coming out, this is like an ultrafiltrate of plasma. And filtrate is due to endothelial lining. So under high pressure, fluid will go out. This fluid which will come out initially, in the very beginning, it will be protein poor. We call it initial process of transudation. Transient transudation. Now, what is transudation? I will explain later that exudation will occur. But transudation is when pressure in microcirculation increases and under hydrostatic pressure, fluid is pushed out in inflamed tissue, and this is ultrafiltrate of plasma, and we call this process transudation, right? Now, but in a very short time, what will happen that endothelium more chemical mediators will come and endothelial cell will shrink and interendothelial gaps will appear. What will appear? Interendothelial gaps. These are interendothelial gaps. As soon as more chemical mediators act on the endothelial cells becomes swollen and their edges retract. And when their edges retract, interendothelial gaps appear. Now fluid will come out more, but this time fluid will come out with proteins, especially small proteins, plasma proteins. So next stage will be that fluid is coming out and this fluid is protein rich fluid, and this protein rich fluid which is coming out, it is due to two mechanisms. Number one, increase pressure in the microcirculation. Number two, increased permeability of microcirculation. Due to increased permeability of microcirculation, and such fluid which is protein rich fluid and it is coming out, this fluid is called exudate. And this escape of protein rich fluid from the vascular compartment into interstitial area or the body cavities, this process of escape of what? Protein rich fluid. This is called process of exudation, and the fluid which is leaking out, this protein rich fluid, is called exudate. So now exudation, after transient transudation, then there will be prolonged exudation. Protein rich fluid will come out. Now you see, as fluid, blood was healthy tissue, blood is moving in a laminar fashion. But here what has happened? Due to vasodilation, more fluid has come, more blood has come here, and due to increased permeability, protein rich fluid has escaped, but yet all the cells are within the vascular compartment. WBCs, leukocytes, red blood cells and platelets are within the circulation, but fluid has gone out. So this will lead to what phenomenon? It will lead to hemoconcentration. This will lead to hemoconcentration, or we can say there is increased viscosity of blood within the microcirculation. Right? There was arterial dilation, increased hydrostatic pressure here. Initially, protein poor fluid came out, that was transudation. Then chemical mediators acted on endothelial lining and interendothelial gaps appear. And when interendothelial gaps appear, microcirculation permeability is increased, and now protein rich fluid come out, we call it exudation. So after a transient transudation, there is prolonged exudation. Now this escape of fluid is there and escape of protein is there, but cells are still behind. This will make the remaining blood in the microcirculation of inflamed tissue, yes, hemoconcentrated, or it will have more viscosity. When it will have more viscosity, it will move slowly as compared to the normal blood flow. That blood is coming from arterial side, normally making some exchanges in microcirculation, giving the nutrients, for example, and removing the waste products, and then blood is moving to the venous side. But here there is too much fluid loss. So this hemoconcentration and increased viscosity make the blood flow slow. So more blood is coming into inflamed microcirculatory area and less blood is leaving due to hemoconcentration and viscosity. So more blood coming in, less blood leaving. So here tissue become, yes, what? Relatively blood flow becomes static. So we say there is relatively some stasis. When there is some stasis, RBCs will aggregate. Red blood cells will aggregate when blood flow is slow. Number one, blood viscosity is high, hemoconcentration is there, RBCs are not properly lifted and they are not repelled properly from each other, and red blood cells or erythrocyte start making small aggregates. Now, when erythrocyte in this area will start making aggregates, then what happens? The aggregates of erythrocytes are larger than individual white blood cells. Now these which were separate from each other, they make aggregates, and aggregates of red blood cell is in size larger than individual leukocytes. So what happens? That they will kick, this red blood cell group will kick on the neutrophils or on the leukocytes, and these leukocytes will be pushed away from the central axial flow. What I'm trying to tell you? I'm trying to tell you that in inflamed microcirculation, where there is hemoconcentration and high viscosity due to loss of fluid as transudate and eventually prolonged exudation process, here become slight stasis, and under these altered hemodynamics of inflamed vasculature, what happens? Red blood cell aggregate become larger than the individual white blood cell. Normally, individual white blood cell is larger than red blood cell. For example, red blood cell is 7 micron and neutrophil is, for example, 12 micron. But when there is a clump of RBCs, it becomes bigger than single WBC. So it will kick on the ass of these centermost cell, and when aggregates of red blood cell will bump with the individual what? Leukocyte, they will kick the leukocytes out of the central flow, and leukocyte will be marginated. They will be lateralized and they will move on the more lateral side. So pattern of blood flow will happen in such a way that neutrophils are now coming on the periphery and they start rolling on the endothelial cells. Right? Now this process of pushing the white blood cell from central axial blood flow in inflamed microcirculation towards the periphery and more near to the endothelial cell is a requirement. It's a requirement that will be followed by expression of adhesion molecules, as we will see in next lecture, that as neutrophils or other white blood cells, when they become near to endothelium, which is activated by different chemical mediators, this endothelium is expressing different molecules like selectins, and selectins will start interacting. Selectins, P selectins and E selectins are on endothelium and L selectins are on white blood cells, neutrophils. These selectins will interact with their corresponding receptors, which I will discuss in the next lecture, and neutrophils will start tethering or transiently sticking, or you can say they will attach under the influence of selectin, but this binding with selectin and their receptor is very weak. they will attach and then detach. They will attach and then detach. It is just like that from the axial flow, they have come to the margin, then they are attaching and detaching with the endothelial cell under the influence of selectins and their ligands. It's just like soft and sweet kisses. This kissing process starts between, what is this, neutrophils and endothelium, right? But I will talk about the sweet kisses, the rolling process mediated by selectins and their ligands in big detail in next session. And then I will talk about that once kissing process is going on, then another group of molecules are activated on the neutrophils, which are called integrins, and their ligands will appear on the endothelium, and those ligands and integrin, they will make very strong bond, because soft and sweet kissing has been done by selectins. Now integrin molecules, which are expressed, not really expressed, they are already present on neutrophil, they are converted into high affinity status, and they interact with their special, I will explain later, receptors on the, I will tell you, these are special receptors, VCAMs and ICAMs, vascular, VCAM vascular cell adhesion molecules, and ICAM intercellular adhesion molecules. These molecules appear on endothelium, and integrins are converted into high affinity state, and they will interact with each other, and kissing has been done under the selectins. Under the integrin activation, they will hug with each other. Now this kissing and hugging I will explain in the next session, and once enough rolling under the selectins or kissing has been done, and then firm adhesion of white neutrophils under the influence of integrins, strong hugging has been done. After that, I will explain in between the endothelial cells, these kissing and hugging neutrophils will, next step is what? After kissing and hugging, penetration. So these neutrophils will penetrate between the endothelial lining, more commonly interendothelial gap, and they will move through that, and this process, and escape from the lumen via the endothelium out of the, you can say, towards the basement membrane here, right? And then eventually to interstitial tissue. This process is called diapedesis, and I will explain also in detail how diapedesis is mediated by PCAMs, platelet endothelial cell adhesion molecules, JAMs, which is basically junctional adhesion molecules, and CD cluster differentiation 99 molecules. These all molecular steps will be explained later. And after that, I will love to teach you about chemotaxis, and I will explain how eventually the neutrophils or macrophages eventually destroy or limit or contain the source of injury or infection, right? Right now, I will just give you a very brief review. First of all, as I told you, that what is margination? A process in which axially flowing, centrally axially flowing white blood cells are kicked out of central flow to the peripheral area towards the endothelium in the inflamed areas microcirculation. This process is called margination. Margination is prerequisite before the adhesion molecules between the endothelial cell and leukocyte interact with each other, and selectins and integrins, they are interacting, and white blood cells become adherent to this endothelium, and they don't leave the circulatory system. They don't simply come and go. Rather, they come from arterial side, they come on the margin, marginated, and later on we'll see there is soft kissing, rolling, and then strong hugging, that is firm adhesions, and eventually passing in between the endothelial cells or sometimes through the endothelial cell, that is diapedesis. All these things we will discuss later. Here I want to just review with you. Do you remember what are different types of chemical mediators which induce vasodilation? Chemical mediators which induce vasodilation. Just I will tell you few chemical mediators which are produced at the site of injury and induce vasodilation. Number one is histamine. Histamine coming from the granules of mast cells in the injured area. Histamine. Then from the cell membranes, lipids of mast cells, resident macrophages and injured cells and even endothelial cell, there is production of prostaglandins and prostacyclins and platelet activating factor. Now prostaglandins also produce vasodilation, prostacyclin also produce vasodilation, and platelet activating factor also produce vasodilation. Along with that, some other vasodilatory substances are produced more in injured tissue like nitric oxide and bradykinin. About the platelet activating factor, I want to put something in your mind. You know, platelet activating factor, even though its name is that it is activating the platelet, but actually platelet activating factor is such a chemical mediator which mediates almost every step of acute inflammation. No need to remember their action, just remember the steps of acute inflammation. The platelet activation can produce, what is this? Vasodilation. Platelet activating factor can produce increased permeability. They can also produce, facilitate adhesion, platelet activating factor. They can also facilitate diapedesis. They can also, platelet activating factor can also mediate as a chemotactic agent, and they can also increase the phagocytic activity and other you can say cidal activity of the neutrophils. So they are powerful neutrophil activator also. So platelet activating factors not only act as vasodilator, they can stimulate every step of acute inflammation, right? So what are the factors which produce vasodilation? I told you histamine, prostaglandins, prostacyclins, nitric oxide, right? And with that, I told you platelet activating factor and bradykinin. And right now we come to what are the different chemical mediators which increase permeability and induce exudation, right? The chemical mediators which can stimulate this exudation and which result eventually what? These chemical mediators will act on the endothelial cell. Endothelial cell will become retract their ends and interendothelial gaps will become wider or appear and protein-rich fluid will come out. What are these chemical mediators? These chemical mediators is again histamine. This is again histamine, which is doing vasodilation as well as this process, increased permeability. Histamine plus leukotrienes, C4, D4 and E4. And again, of course, platelet activating factor working here, and also interleukin 1, tumor necrotic factor, nitric oxide work here as well as here, bradykinin work here as well as here, right? So these were different chemical mediators. All these chemical mediators, by mediating the vasodilation and by increasing the permeability of the microcirculation, lead to hemoconcentration and viscosity and congestion in this area, and eventually these changes lead to red blood cell aggregates which kick the individual white blood cells to the margins and they bump into what? Endothelial lining. And then what happens? We will discuss next time. Here I want to make one more concept clear, that in this inflamed area, now there is, because of vasodilation, there is more blood present, right? And more blood is coming and less blood is leaving due to hemoconcentration and viscosity. So total amount of blood which is present in vasculature of inflamed tissue is more than normal, right? So this is said that tissue vasculature, microvasculature is engorged. What is this? Engorged. Now, what is engorgement? What is hemostasis? Not homeostasis, forget about it. Just say when microcirculation is engorged, right? This is a situation where there is more than normal blood is present in microcirculation in the inflamed tissue. Now, this engorgement is due to two processes. Number one, increased inflow due to arteriolodilatation and decreased outflow of blood due to hemoconcentration and stasis and high viscosity. So both of them are, so more blood coming here, less blood leaving the inflamed tissue, both of them are leading to engorgement, right? But when there is more blood in microcirculation, mainly due to arteriodilatation, we say there is hyperemia. What is that called? Hyperemia. So what is hyperemia? Hyperemia is a condition in which there is excessive blood present in microcirculation, right? And microcirculation is engorged with the blood, and primary mechanism is due to arteriolodilation, right? But if in some tissue microcirculation engorgement is primarily due to decreased outflow. For example, if there is some venous obstruction, here there is not physical venous obstruction, here is viscosity and hemoconcentration. But just imagine that if in some tissue there is outflow obstruction here. So blood is coming here but unable to leave properly, and blood start accumulating here. This situation where microcirculation is engorged because venous outflow is reduced, this is called congestion. What is it called? Congestion. So congestion is such engorgement in the microcirculation where venous outflow is reduced. Hyperemia is such engorgement of microcirculation where inflow of blood is more. And in inflammation, hyperemia and congestion both occur, right? In inflamed area, hyperemia and congestion both occur. Here we'll take break, and after the break, we'll talk about this soft kissing, rolling by via selectins, and strong hugging, firm adhesions by integrins, and eventually penetrations in between the endothelial cell to come out of the vascular compartment, diapedesis, which is mediated by PCAM1s and JAMS molecule and CD99. So let's have a break. After that we continue. Thank you very much.